Snake eyes have been watching the world for over 100 million years, and they work nothing like yours. There's quite a lot going on behind that fixed, unblinking gaze. Snakes have no movable eyelids. Each eye is permanently sealed beneath a single transparent scale called the brill, sometimes also called the spectacle, which is fused directly over the cornea. The snake cannot blink, cannot wink, cannot ever close its eyes, not even in sleep. Every moment of a snake's life, its eyes are fully open to whatever world surrounds it. That brill is shed along with the rest of the skin during the snake's periodic molt, a process called ecdysis. In the days just before shedding, fluid accumulates in the thin space between the brill and the underlying cornea. The eye turns a milky, clouded blue, a phase keepers of captive snakes know well. For that brief window, which can last several days, the snake's vision is genuinely impaired. A temporarily half-blind predator, waiting to renew itself. Unlike mammalian eyes, which focus by changing the shape of a flexible lens, flattening it for distance, rounding it for close objects, a snake focuses by physically moving the lens forward and backward within the eye. The lens shifts position rather than deforming. It is a different mechanical solution to the same optical challenge that every vertebrate eye has needed to solve, and snakes arrived at their own answer independently of the path mammals took. If this kind of science interests you, a like and subscribe helps this channel reach more people. Have you ever looked closely at a snake's eye and found it impossible to tell exactly where it was looking? That experience connects to something structural. The iris of a snake contains striated muscle rather than smooth muscle, the same kind of fast-acting, high-force muscle that moves limbs, instead of the slow-twitch smooth muscle that controls pupil size in most other vertebrate eyes. Striated muscle contracts faster and with considerably more force. The result is a pupil capable of extremely rapid, powerful constriction and dilation. One that can snap from a wide, light-drinking opening to a hairline slit almost instantly. Pupil shape also varies dramatically across the roughly 3,700 known snake species, and it tracks ecological lifestyle closely. Nocturnal hunters tend to have vertical slit pupils, which allow extreme dilation in darkness while still permitting rapid constriction in bright daylight. Daytime hunters more commonly have round pupils. And certain arboreal rearfang species, tree snakes that spend their lives above the ground, have horizontal or keyhole-shaped pupils, a configuration thought to improve their view of the branches and ground below them, giving a wider effective field along a plane parallel to the surface they perch above. Most snake retinas are dominated heavily by rod photoreceptors rather than cones. Rods are sensitive to light intensity and to motion, rather than to color. The practical consequence is that most snakes perceive the world with limited color discrimination but with considerable sensitivity to movement, even very subtle movement, even at low light levels. A flickering mouse in near darkness is far more visible to a snake than a perfectly still one at close range. This is not a limitation of the visual system so much as a feature of it. The eye was shaped by what the animal needed to find. A good companion for the quieter hours, the kind of science the mind can move through slowly. Did you ever notice how different snake eye colors look between species? Snake eyes come in a wide range of colors: gold, copper, pale silver, flat black, and even red. These colors arise from pigmentation in the iris rather than from the structure of the eye itself. Broadly, golden or reddish irises appear more often in diurnal species, where warmer iris pigments may reduce the amount of scattered light entering from the side, improving contrast. Darker irises dominate in nocturnal snakes, where absorbing excess daytime light matters less than gathering every available photon after dark. The eye color you see is not decoration. It is the outcome of pressure from the light environment the animal lives in. The cornea of a snake is not protected by an eyelid the way mammal corneas are. The brill substitutes for this, but it is entirely passive, no active cleaning mechanism, no sweeping motion, no tear film spread by blinking. Snakes also have no tear ducts in the mammalian sense. Any fluid associated with the eye drains internally through a duct that opens into the mouth, not outward over the face as tears do in most vertebrates. It is one of the stranger plumbing arrangements in vertebrate anatomy, and it means that what can look like a teary snake eye is usually something else entirely. The way visual detail is distributed across the retina also varies by how a snake makes its living. Some species have a horizontal streak of elevated retinal ganglion cell density running across the middle of the retina. This concentrates the highest resolving power along a wide horizontal band of the visual field, precisely the region that sweeps across flat terrain where low, fast-moving prey would appear. The retina is not a passive sensor. It is already making decisions about what matters before the signal ever reaches the brain. The lens of most snakes is nearly spherical. A spherical lens gathers more light than a flatter one, making it well-suited to low-light conditions. The trade-off is reduced ability to independently resolve objects at very different distances in bright light. This is a compromise, not a flaw, and it reflects a history spent in environments where gathering light mattered far more than isolating a nearby fly from a distant branch. Even in snakes that now hunt in full daylight, the spherical lens remains, a structural memory of a dimmer past. And then there is the question of just how old and how varied the snake eye really is. The oldest surviving snake lineages, a group called Scolopophidians that includes blind snakes and thread snakes, have eyes so reduced that they can do little more than detect light from dark. They are vestigial structures, shadows of what an eye can be. In contrast, the more recently diversified families, colubrids, vipers, elapids, developed substantially more complex optical systems, rebuilt from a simplified ancestor over tens of millions of years. The snake eye was not always the refined thing it became. It was, in a real sense, reinvented. There is still one aspect of snake vision that most people have never considered, something about the way the brain handles what the eye collects. And that opens into a different kind of story entirely. What looks like a simple, cold stare is already, even at the level of basic anatomy, a system with a very long and surprisingly intricate history behind it. The pupil is one of the most information-dense features on a snake's face, and also one of the most misread. Consider the vertical slit. When a snake with slit pupils is in dim light, the pupil opens wide, sometimes filling nearly the entire visible eye. But when bright light hits it, the iris constricts that slit down to an almost invisible hairline line, a narrow dark dash cutting through the iris. What makes this configuration so useful is the range it covers. A round pupil that dilates and constricts has a limited ratio of maximum to minimum area. A vertical slit, because of the geometry of how the two edges close toward each other, can achieve a far greater range between its widest and narrowest states, allowing the same eye to operate efficiently across a much wider spread of light intensities, from deep forest shadow to direct midday sun. Round pupils tell a different story. Species with round, fully circular pupils, green tree pythons in morning light, many of the active hunting colubrid snakes that move across open ground during the day, tend to be diurnal foragers. The round pupil suits an eye that isn't managing extreme swings between bright and very dark conditions, and it pairs commonly with a retina that has a higher proportion of cone photoreceptors, supporting somewhat better discrimination of color or fine detail during reliable daylight hours. The round pupil is not simply less specialized than the slit. It is specialized for different conditions. Then there are the horizontal pupils, rarer, and found in particular arboreal species like some of the rear-fanged colubrids in the genus Ahatula, the Asian vine snakes. In these snakes, the pupil elongates along a horizontal axis, and a groove along the snout ahead of the eye extends the effective downward visual field. The configuration is thought to help the snake look past its own body toward the ground or toward a branch below it. Arboreal life asks specific visual questions. Where does the next surface sit? How far away is the prey item on that thin twig? The horizontal pupil may be one part of the eye's answer to those particular questions, though research into its exact functional advantages is still developing. In some species with deeply slit pupils, the constriction in bright light is so extreme that the pupil effectively disappears from casual observation. The slit narrows until the eye looks nearly featureless from a short distance, a dark iris with no obvious opening visible. The snake can still see clearly through this nearly closed aperture. The optics still work because even a very narrow aperture transmits enough light for the sensitive retina beneath it. But to an observer, the eye takes on that flat, unreadable quality that has fed so much folklore about snake vision. The variety of pupil shapes across snake species is wide enough that researchers studying it in depth have had to develop systematic classification schemes just to describe it consistently. Among the roughly 3,700 recognized snake species, a number that continues to shift slightly as taxonomy is revised, pupil form ranges from round to vertical slit to horizontal oval to teardrop to the more unusual subtriangular shapes seen in a few rear fang specialists. This diversity reflects not a single evolutionary solution but a history of repeated, independent modifications to the same basic ocular structure, shaped by the pressures of each ecological niche a snake lineage moved into. The cat-eyed snakes of the genus Boiga are worth pausing on. These are rear-fanged colubrids, largely nocturnal, found across South and Southeast Asia. They have vertical slit pupils, as expected for nighttime hunters. But their eyes are disproportionately large for their body and head size, noticeably large even to casual observation. The combination of a wide aperture slit and a large eye means the total light-gathering capacity of the Boiga eye is substantial. These snakes hunt lizards and birds in trees after dark, and the eye appears scaled to exactly that challenge. Big enough to catch what little light the canopy passes, with a pupil capable of snapping near shut by dawn. Now, here is something worth correcting because it comes up frequently. Pupil shape in a snake is not a reliable indicator of whether that snake is venomous. The idea that vertical slit pupils mean dangerous and round pupils mean safe has circulated widely and persistently, particularly in field guides and online identification resources. It does not hold up. Many highly venomous species have round pupils. King cobras, black mambas, many taipans. Many completely harmless snakes have vertical slits. Pupil shape is a product of activity pattern and light environment, not toxicology. Relying on it as a safety assessment in the field is genuinely hazardous. The herpetological literature, including work published in journals like Copy.ia and the Journal of Herpetology, consistently treats this as a debunked correlation rather than a useful rule. Back to the mechanics. The degree of pupil asymmetry between the two eyes, meaning a situation where the left and right pupils are not exactly the same size or shape under identical lighting, has been observed in some snake species. This is not the norm. Under most conditions, the two pupils respond in coordination. But under certain experimental lighting conditions, and occasionally under stress or in the presence of pathology, mild asymmetry appears. Its functional meaning is not well established. It may be a subtle artifact of differential neural inputs under unusual conditions, or it may reflect mild differences in the muscular or neural apparatus of each eye. The honest answer is that this has not been thoroughly studied in most species. That striated iris muscle, mentioned in the anatomy of the snake eye, also has another consequence worth noting here. The force a snake's iris can generate in full constriction is measurably high relative to the iris muscles of vertebrates with smooth muscle controlling their pupils. In bright sunlight, the iris of a strongly photophobic species can constrict tightly enough to produce what looks, under close examination, like a nearly occluded aperture. The pupil does not disappear entirely. That would require a complete seal of tissue that biology hasn't arrived at. But the opening can be reduced to a fraction of a millimeter in some individuals under full direct sunlight. That is an extreme degree of active constriction. The pupillary light reflex, the automatic constriction response to a bright light stimulus, is present across snake species but operates with different speeds. Snakes with striated iris muscle tend to show faster reflexes than would be typical of a smooth muscle-controlled iris. The difference is not trivial. Striated muscle responds to its neural signals in milliseconds, while smooth muscle contracts over a longer time window. For an animal moving between patches of light and shadow, or hunting prey that appears briefly in a gap in the canopy, faster pupil response means faster visual readjustment. The striated iris is not just a curiosity of anatomy. It is a functional advantage in exactly the environments where many snakes operate. Pupil shape, then, is one of the most readable external expressions of a snake's hidden evolutionary biography. The slit says something about a history lived in variable light. The round pupil speaks to a life measured in daylight hours. The horizontal keyhole encodes an arboreal lineage with specific spatial problems to solve. Read carefully enough, the pupil alone tells you something real about where that animal came from, what it hunts, and when its ancestors were most active, even before you know the species name or see another scale on the body. What the pupil cannot tell you, and what turns out to involve an entirely separate sensory system, is waiting further along. Knowing the structure of the eye is one thing. Knowing what it actually delivers to the animal behind it is something else. Most snakes are functionally dichromatic, meaning their color vision is built around two types of cone photoreceptors rather than the three that human eyes use. Each cone type is tuned to a different range of wavelengths, and the brain compares the signals from both to derive a sense of color. With two types instead of three, the color world a dichromatic snake inhabits is narrower than ours. Fewer distinctions available between colors that sit close together in the spectrum. A blue-green surface and a yellow-green surface that look clearly different to a human eye might appear much more similar to many snake species. That said, dichromacy is not color blindness in any simple sense. It is a different window onto the spectrum, not a shuttered one. Some snake species are sensitive to ultraviolet light, wavelengths shorter than violet, entirely invisible to human eyes. This UV sensitivity has been confirmed in certain colubrid species through electrophysiological recordings and behavioral testing. The functional significance of UV detection in snakes is still being worked out. One hypothesis connects it to tracking prey. Some small mammals, particularly rodents, have urine that reflects UV light. A snake capable of detecting UV might be able to follow scent trails that also have a visible component, a kind of cross-modal confirmation. The evidence for this specific use is suggestive but not yet definitive. Motion is the dominant trigger for most snake predation events. A mouse sitting perfectly still within striking range of a hungry rat snake may remain entirely undetected for an extended period. The moment it moves, even slightly, even briefly, the snake orients and engages. This is not poor vision in any simple sense. It reflects a retina shaped by evolutionary pressure to detect exactly what a hunting snake needs to detect. A living thing, distinguishing itself from background by moving. Stationary objects that are not prey have no relevance to the animal, and the visual system has not been organized around cataloging them in detail. Have you ever watched a snake's head track something moving across the ground? That deliberate, perfectly smooth tilt of attention? That motion sensitivity is not uniform across the visual field. Some species show higher motion detection sensitivity in specific regions of the visual field that align with where prey typically appears given their hunting method. Ground hunting species often show more sensitivity along the horizontal band, close to the substrate. Species that take birds show more sensitivity in the upper field. The retina is not just detecting motion. It is sampling different regions of space with different priorities baked into its cellular architecture from the beginning. Among diurnal snake species, some show a meaningfully higher ratio of cone photoreceptors to rods compared with their nocturnal relatives. This shift toward cone-dominated vision supports better performance in the specific tasks that bright light environments demand: color discrimination, fine edge detection, resolving detail in high-contrast scenes. Certain colubrids that hunt by sight in open environments during the day appear to have pushed toward this end of the spectrum, developing retinas that sacrifice some low-light sensitivity in exchange for improved daytime performance. The retina across snake species is not a fixed design but a tunable system that evolution has adjusted repeatedly. The optic tectum, a structure in the midbrain that processes visual input and coordinates orienting responses, is proportionally larger in visually active, sight-hunting snake species than in those that rely less on vision, such as fossorial burrowers. This pattern across species connects brain anatomy directly to behavior and ecology. Where vision is the primary hunting tool, the visual processing infrastructure expands. Where it is secondary or vestigial, the structure shrinks. This relationship between brain region size and ecological reliance on that sense appears across vertebrates broadly. Snakes are a particularly clear example because the variation across species is so large and so strongly tied to lifestyle. True visual acuity, the resolving power that distinguishes fine detail, that lets you read text or identify a face at a distance, is considerably lower in most snakes than in humans. The snake retina does not need to read text. It needs to detect a moving, warm-blooded animal against a background of leaves, and to do that from a position of ambush or pursuit. Resolution in the human sense is simply not what the system was built for. Measured acuity in tested species is typically low enough that the image projected onto the snake retina would look blurry to us by comparison, though to the snake, that image contains the information it actually needs. Most snake species also lack a true fovea. The fovea is the small central region of the human retina packed densely with cone cells, responsible for the sharp central vision that allows reading and face recognition. Some snake species have a fovea-like depression, a shallow pit where photoreceptor density increases slightly compared with the surrounding retina, but it does not reach the extreme cone density of a true fovea. The visual world of most snakes is broadly, somewhat uniformly sampled rather than sharply centralized, with a center of high resolution surrounded by lower resolution peripheral vision the way the human eye works. Certain arboreal snakes have eyes that face more forward than the strictly lateral positioning common in most species. Forward-facing eyes produce more overlap between the fields of view of the left and right eyes, a wider binocular zone. Binocular vision, where both eyes receive slightly different images of the same object, allows the brain to calculate depth from the disparity between those two images. This is the same principle that underlies human depth perception. For a snake living in three-dimensional tree canopy, judging the exact distance to a thin branch or a perched lizard has real survival value. The eyes, over evolutionary time, have moved to make that calculation more precise. Signals from both eyes partially converge in some snake species in the optic tectum, providing the neural substrate for at least a rudimentary form of binocular processing. This is not the full stereoscopic vision that primates use. The convergence and the dedicated neural machinery are less elaborate, but it is more than the purely monocular processing of species with fully lateralized eyes. The degree of convergence varies across lineages and appears to scale with how forward-facing the eyes are, which scales in turn with how arboreal and visually oriented the lifestyle is. Head bobbing, that rhythmic forward and back or side-to-side movement seen in certain snake species when they appear to be assessing their environment, has been interpreted by researchers as a motion parallax strategy. Motion parallax is the apparent difference in how fast near objects and far objects seem to move when the observer moves. By generating its own head movement, the snake creates apparent motion in the visual scene that it can use to judge relative distances, even without binocular overlap and without a fovea. It is a behavioral compensation for optical limitations, generating information that the eye alone cannot deliver by moving through space. And snakes do not appear to use smooth pursuit eye movements of the kind that allow humans and many other vertebrates to keep a moving object locked in the center of the visual field as it travels across the scene. The snake eye does not swivel in its socket to follow prey smoothly. Instead, the snake moves its whole head, reorienting the entire visual apparatus rather than redirecting gaze within it. This is partly anatomical. The range of eye rotation available in the socket is limited in most species. The snake tracks the world by turning itself toward it rather than by redirecting its gaze within a fixed head. There is a layer beneath all of this visual processing, one that operates entirely outside the range of light, that changes everything about how some snakes experience the world around them. That system has its own distinct biology, its own evolutionary history, and its own deeply unusual neuroscience. Some snakes do not just see the world. They feel it as a thermal image, painted in heat rather than light. The L'Oreal pit organs of pit vipers, rattlesnakes, copperheads, cottonmouths, and many of their relatives are small, membrane-lined cavities positioned between the eye and the nostril on each side of the face. These are not nostrils. They are not accessory eyes. They are dedicated infrared detectors, each one consisting of a thin, suspended membrane stretched across a fluid-filled chamber, capable of responding to temperature changes in the environment in front of the snake. Each pit has an outer chamber open to the air and an inner chamber connected to the environment through a small duct, and the membrane between them is the actual sensing surface. The arrangement is delicate, specialized, and unmistakably purposeful. pythons and many boas have a different configuration. Rather than a single L'Oreal pit on each side of the face, these snakes have rows of small pit organs arranged along the lip scales, labial pits, embedded in the supralabial and infralabial scales that border the mouth. The organization is different from the pit viper system, and the two evolved independently. When the same functional solution appears in separate lineages without shared ancestry for that trait, it is called convergent evolution, and the heat-sensing pit organ is one of the cleaner examples of convergence in snake biology. The same engineering answer arrived at twice through entirely different routes. At the molecular level, the pit organ membrane is lined with a dense mesh of free nerve endings expressing a protein called TRPA1, a transient receptor potential ion channel. This same channel is found in the pain and irritant sensing pathways of mammals, including humans, where it responds to things like cold temperatures, chemical irritants, and certain compounds in mustard and wasabi. In the pit organ membrane of pit vipers, TRPA1 has been co-opted into an infrared detector, responding to the radiant heat of warm objects rather than to chemicals. The molecule itself is ancient and widespread. The snake did not invent it. It redirected a pre-existing sensor toward a new problem. Research published in Nature in 2010 by Gracheva and colleagues identified TRPA1 as the molecular basis of pit organ infrared sensitivity, which resolved a long-standing question about the cellular mechanism involved. The sensitivity of the pit organ membrane is genuinely extreme. Studies of pit viper species have measured responses to temperature differences as small as around 0.003 degrees Celsius, fractions of a degree in other words. The membrane does not need the warm object to touch it or to be especially close. It detects radiant infrared energy, heat traveling through air as electromagnetic radiation, from objects at behavioral distances. A mouse moving through darkness near a rattlesnake radiates a continuous thermal signal that the pit membranes are registering and updating in near real time. Here is where the system becomes neurologically unusual. The infrared signals captured by the pit organ membranes travel via branches of the trigeminal nerve, not the optic nerve, to the brain. And they are processed not in a separate dedicated region but in the optic tectum, the same midbrain structure that handles visual input from the eyes. The result is that the brain receives two spatially organized maps of the environment simultaneously, one built from light, one built from heat. These maps overlap in the same neural tissue. The snake is not choosing between seeing and heat sensing. It is integrating both into a single coherent representation of the space in front of it. Something worth clarifying directly, because it surfaces often in popular descriptions of these animals. The pit organ is sometimes described as a kind of heat vision, as if the snake sees infrared light the way a thermal camera displays it. The reality is more constrained than that. The spatial resolution of the pit organ is considerably lower than true vision. The pit opening acts as a crude aperture, and the membrane samples a broad area of incoming infrared radiation rather than resolving fine detail. What the pit delivers is approximate location, direction, and movement of warm objects, not a sharp thermal image. A rat in darkness is detectable as a warm, moving presence with a rough angular position. It is not rendered in detail. The precision of the system is sufficient for strike targeting at close range, but it is not the high-resolution thermal imaging that cinematic representations of snake vision often suggest. The kind of science that carries the mind gently outward through biology, through nervous systems, through the long quiet history of animals solving problems in the dark. Even so, The behavioral consequences are not modest. Strike accuracy studies on pit vipers hunting in complete darkness show that strikes are directed at the prey's body location with meaningful precision, not randomly into the dark, not wildly off target. The accuracy is not as high as under lighted conditions with both systems active, but it is well above chance. The pit organ alone, operating without any visual input, is sufficient to support a functional predatory strike. An animal that evolved this system gained the ability to hunt through the full night, through total underground darkness, through any environment where light is absent but warm prey is present. Elapid snakes, the family that includes cobras, mambas, krites, and taipans, do not have pit organs at all. Some of the most medically significant venomous snakes in the world, animals with highly developed venom delivery systems and documented predatory efficiency, hunt entirely without infrared sensing. They rely on vision, olfaction, and vomeronasal input. The pit organ is not a prerequisite for being an effective predator among snakes, or even for being venomous. It is one tool that some lineages developed, not a universal feature of advanced snake biology. The free nerve endings in the pit membrane are supplied so densely that the membrane itself has been described in anatomical literature as among the most richly innervated tissue found in any vertebrate sensory organ relative to its area. The trigeminal nerve branches serving the pit are enlarged in pit viper species relative to non-pit-bearing relatives. The neural infrastructure has been scaled up to match the sensory demand. The body has not simply added a membrane and left the wiring unchanged. It has rebuilt the relevant portion of the peripheral nervous system around the new sensor. In the boas and pythons with labial pit organs, the functional output is similar to pit vipers, infrared sensitivity sufficient for prey detection and strike targeting, but the anatomy of the pit itself is simpler. The labial pits lack the double-chamber arrangement of the L'Oreal pit. The membrane is still sensitive, still trigeminal nerve connected, still feeding into overlapping visual thermal brain processing. But the structural elegance of the L'Oreal pit, that suspended membrane between two fluid-separated chambers, was not what evolution arrived at in these lineages. Two solutions, both functional, both independent, both using the same ancient molecular tool at the cellular level. The pit organ also raises a question that the eye alone cannot answer. Which of the two systems does the snake weight more heavily when both are providing information simultaneously? In well-lit conditions with visible prey, behavioral evidence suggests vision dominates. Snakes with both systems respond faster to visual targets when light is present. In darkness, the pit organ takes over entirely as the primary orienting system. In low-light conditions where both systems are providing partial information, the integration appears to be additive. Accuracy improves over either system alone. The brain, it seems, treats the two maps as complementary rather than redundant, combining them rather than choosing between them. What the snake has built across millions of years and across multiple independent lineages is a sensory system that refuses to be limited by the availability of light. The eye works when light is present. The pit works when it is not. Together, they cover the full range, a perceptual toolkit that reaches from the visible spectrum all the way into the thermal infrared, drawing on two completely different physical phenomena to build one picture of the world. Every vertebrate eye faces a version of the same problem. The surface of the eye is exposed to the world, and the world is full of things that damage surfaces. Most vertebrates solve this with eyelids, movable flaps of skin that sweep across the cornea, distribute moisture, remove debris, and close during sleep or threat. Snakes went a different direction entirely. The brill, that single fused transparent scale permanently sealed over each eye, is not a simplified eyelid. It is a different solution built from different embryological material, producing a different set of capabilities and trade-offs that shape nearly every aspect of how a snake interacts with its visual environment. The brill does not develop from a migrating skin scale that happened to stop over the eye. It forms embryologically from the fusion of the upper and lower eyelid primordia, the same tissue that would have become separate, movable eyelids in a lizard ancestor. During snake embryonic development, those two tissue buds grow toward each other and fuse into a single transparent window rather than forming two independent flaps. The eye is sealed not because a scale grew over it, but because the eyelids merged into one and became optically clear. That developmental origin matters. It means the brill is, in a genuine embryological sense, the snake's eyelids just permanently joined and transformed. In a healthy snake that has recently shed, the brill is optically clear enough that its refractive index, the degree to which it bends incoming light, is close to that of the cornea directly beneath it. The two surfaces behave almost as a single optical unit. Light passes through the brill and into the cornea with minimal distortion or scatter. To an observer, the eye simply looks glossy and unobstructed. The brill is effectively invisible as an optical element under those conditions, which is why it is so easy to forget it is there at all. What makes it unmistakable, and what every keeper of captive snakes knows to watch for, is what happens in the days before a shed. Fluid accumulates in the thin subspectacular space between the brill and the cornea below it. This fluid layer, thin as it is enough to scatter light significantly. The eye clouds over, turning a blue-gray or milky color. The snake enters what is colloquially called the blue phase, or being in the blue, a period of genuinely compromised vision that can last several days. During this window, many snakes become noticeably more defensive or withdrawn, which makes biological sense. An animal that knows its vision is impaired responds with appropriate caution. Have you ever watched a snake just after a fresh shed? That moment when the old skin peels away and everything underneath looks suddenly new? After a complete, successful shed, the old brill lifts away with the rest of the skin as a single, continuous piece. The new brill beneath it is fully formed and clear. Vision returns quickly. Within hours of a clean shed, the optical system is operating normally again. It is a kind of renewal that has no direct parallel in mammalian biology, where the cornea is maintained continuously rather than periodically replaced wholesale. The snake solution is periodic rather than continuous maintenance. Accept temporary impairment, then reset completely. A shed that fails to come away cleanly at the eye produces a condition called retained spectacle. One or more old brill layers left behind over the eye instead of peeling free with the skin. In captive snakes, this is a recognized veterinary problem. A single retained layer may cause no immediate harm, but multiple retained layers accumulating over successive sheds can impair vision progressively, create a physical pressure on the underlying eye, and trap moisture in the subspectacular space in ways that increase the risk of bacterial infection. Veterinary treatment involves careful rehydration and, in persistent cases, gentle manual assistance with removal, a procedure that requires considerable care because the brill and the cornea beneath it are separated by only that thin fluid space. Forcing a retained brill off a dry eye risks pulling corneal tissue with it. The brill has no secretory glands of its own. It does not produce any kind of surface fluid, does not maintain itself biochemically, and has no active cellular maintenance mechanism during the interval between sheds. Whatever moisture surrounds the underlying cornea during the intershed period comes from the internal tissues, diffusing outward through the subspectacular space. The brill is, from a maintenance standpoint, entirely passive between sheds. The snake cannot top up its corneal moisture by blinking, cannot flush the surface with a fresh tear film, cannot clear a speck of debris mechanically once it lands on the brill. The only cleaning the brill receives is the total replacement at each shed cycle. Because the eye cannot blink and cannot close, there is also no mechanical protection from sudden impacts. The brill provides a physical barrier. It is a tough, keratinized structure, but the eye beneath it is always exposed, always forward-facing, never sheltered by a closing lid the way a mammal can reflexively shut its eye against an approaching object. Snakes do exhibit a withdrawal response when objects approach the eye rapidly, pulling the head back or tucking it, but the eye itself has no active mechanical shield beyond the brill it was born with. In some species, the brill is not perfectly flat but carries a slight outward curvature. A curved transparent surface adds its own small refractive contribution to the optical system, effectively a weak additional lens sitting in front of the cornea and the crystalline lens behind it. The optical contribution of a mildly curved brill is small compared with the power of the main crystalline lens, but it is not zero. In species where the curvature is measurable, it adds marginally to the eye's total light-bending capacity. Whether this represents a functional adaptation or simply a structural consequence of how the fused eyelid tissue settles during development is not fully resolved. Infections of the subspectacular space, the cavity between the brill and the cornea, are documented in captive snake medicine as subspectacular abscesses. The infection typically presents as a visible cloudiness, bulging, or discoloration of the eye that differs from the uniform haziness of pre-shed fluid accumulation. In the wild, these infections are rarely documented, partly because wild snakes with significant eye pathology are less likely to be observed and partly because regular veterinary monitoring of wild populations is not routine. In captivity, treatment may involve careful drainage of the subspectacular space, sometimes combined with systemic antibiotic treatment. Left untreated, severe infections can cause permanent damage to the underlying cornea or deeper eye structures. What the brill ultimately represents, taken as a whole, is an unusual evolutionary consolidation, a single passive structure that simultaneously performs the functions that in most vertebrates are distributed across a corneal surface, a continuous tear film, two movable eyelids, and the debris-clearing sweep of a blink. All of that has been folded into one fused, transparent, periodically replaceable scale. It sacrifices the continuous, active maintenance of the mammalian eye for the simplicity and protection of a sealed, unbroken surface. For an animal that spent a significant portion of its evolutionary history underground, pressing its face against soil and debris, that sealed surface may have been a better solution than any number of movable parts. The brill closes off the eye from the outside world completely. What happens next, inside the eye, in the retina, in the optic pathways, in the brain regions that handle what the eye collects? That is where the story of how snakes actually use their visual system takes on its full shape. To understand why snake eyes are the way they are, you have to go back, much further back than the snakes themselves. Snakes descended from lizard ancestors. That much is established. But which lizards, living how, in what kind of environment? These questions have been contested in the scientific literature for decades, and the debate has real consequences for how we interpret the snake eye's current design. The leading hypothesis, supported by both molecular phylogenetics and certain fossil evidence, proposes that the ancestors of modern snakes became burrowing animals, fossorial subterranean, sometime in the Cretaceous period, roughly 100 million years ago, perhaps more, spending a life underground, pressing through soil and root systems in near or total darkness, exerts very particular pressure on a visual system. The eye that served a surface-dwelling lizard well becomes a liability underground, vulnerable to abrasion, costly to maintain, and largely useless in an environment with no light to gather. An alternative hypothesis, less widely accepted but still present in the literature, suggests that early snake ancestors were aquatic rather than burrowing, and that the eye's simplification reflects an aquatic phase rather than a fossorial one. Both the aquatic and fossorial hypotheses agree that early snakes underwent significant reduction of the visual apparatus. They disagree about the selective environment responsible. The debate remains genuinely open, and honest accounts of snake evolution have to acknowledge that the origin story is not fully settled. Work by researchers including Michael Lee and colleagues has examined this question through both morphological and molecular evidence, without reaching a final consensus that the field has uniformly accepted. The clearest living evidence for extreme visual reduction in an early snake lineage comes from the Scolophagians, blind snakes, thread snakes, and their relatives. These are the most ancient surviving snake lineages, small burrowing animals that spend nearly their entire lives underground. Their eyes are vestigial in a precise biological sense. The structures are present but greatly reduced in size, often partially or fully covered by head scales, and capable at best of crude light versus dark detection. There is no image formation, no motion tracking, no color processing. The eye persists as a developmental remnant. The genetic program to build an eye is still running, but the result is a shadow of what the ancestral lizard eye was. What happened next, in the more derived snake families, the ones that eventually returned to surface life, is one of the more unusual sequences in vertebrate eye evolution. Rather than retaining a complex eye from their lizard ancestors, these lineages appear to have rebuilt eye complexity from the simplified fossorial baseline. The ancestral snake had already lost the elaborate optical and cellular machinery of the lizard eye. The surface-returning lineages had to reconstruct it or approximate it from reduced starting materials. This process is sometimes called secondary elaboration, the re-expansion of a system that had previously been simplified under different selective pressures. One of the most telling signs of this rebuilding history is what snakes are missing compared to lizards. The annular pad, a ring of cells surrounding the lens in lizard eyes that assists with accommodation, the fine-tuning of focus, is absent in snakes. So are several other accessory structures present in lizard retinas. These are not things that snakes never had. They are things that the snake lineage had and lost and did not fully recover. The rebuilt eye works, it clearly works well in many species, but it was assembled from what remained after the fossorial reduction, not from the full lizard toolkit. The cone photoreceptors of modern snakes carry a particularly striking mark of this history. In most vertebrates, rods and cones are distinct cell types with distinct ancestry. Rods for low light sensitivity, cones for color and detail in bright light. In snakes, the cones appear to have evolved not from ancestral cone cells directly but from ancestral rod cells, a process called transmutation, documented through detailed molecular and histological analysis. The implication is that snakes lost their original cone population during the fossorial phase, then rebuilt color-detecting capacity using cells that started out as rods. It is an improvised reconstruction, and it explains certain unusual biochemical properties of snake cone cells that distinguish them from the cones of other vertebrate groups. This finding, developed substantially through the work of researchers like Kate Bott and Gordon Walls, reframed how biologists understand the entire history of the snake retina. The brill, too, may be interpreted through the lens of this burrowing ancestry. Fused eyelids that form a sealed, tough, transparent cover are considerably better suited to a life spent pushing through abrasive substrate than movable eyelids that could catch debris, tear, or fail to close fully under soil pressure. The brill is not just a simplification of the eyelid system. It may be an active adaptation to the fossorial environment, a structural change that increased survival during the underground phase and then persisted, carried forward into every descendant lineage even as the animals returned to entirely different environments. The spherical lens tells a similar story. A highly curved, nearly spherical lens is excellent for gathering light in dim conditions. The geometry maximizes the solid angle of light collection. Underground or in any dim environment, this matters. Lizard lenses are flatter, optimized more for the bright light performance of diurnal surface life. Snake lenses appear to have been rounded during the fossorial phase and retained that shape even in lineages that later became strongly diurnal. The lens is, in this reading, another structural memory, a fossil of the underground life embedded in the optics of animals that now hunt in full daylight. Among the more recently diversified snake families, and especially within the colubrids, which represent more than half of all living snake species, the range of eye complexity is wider than in any other snake group. Some colubrids have eyes that are relatively simple and motion-sensitive. Others have highly developed visual systems with forward-facing eyes, fovea-like retinal regions, and behaviors that depend on precise visual assessment. This spread of visual complexity within a single family reflects the speed and breadth of the colubrid radiation. These snakes moved into nearly every terrestrial and arboreal niche available, and their eyes diversified accordingly. Each new ecological context asked different visual questions, and the eye answered each time with whatever modification the available genetic toolkit permitted. The snake eye is therefore not one thing that evolved once and stabilized. It is the product of a long, interrupted, redirected history, a lizard eye simplified by burial, partially rebuilt by return to the surface, diversified repeatedly as descendants spread across environments, and constrained throughout by the structural decisions made during that first long underground phase. Every feature discussed so far, the spherical lens, the brill, the transmuted cones, the striated iris muscle, the absence of an annular pad, carries that history inside it, readable if you know what to look for. What that rebuilt eye does in darkness, how the nocturnal lineages pushed it to its absolute limits, is a different kind of story, and one that the anatomy of the eye makes considerably more dramatic than it might first appear. Darkness is not an obstacle for many snakes. It is the environment they were built for. The nocturnal snake eye is not simply a diurnal eye with the brightness turned down. It is a different configuration, a set of structural choices made across evolutionary time that prioritize the gathering of scarce photons above almost everything else the eye could alternatively do. Understanding those choices and what they cost is one of the cleaner ways to see how biological trade-offs actually work in practice. Begin with the pupil. In strongly nocturnal species, the pupil at full dilation occupies a larger proportion of the total eye diameter than in diurnal counterparts. The opening is wide not merely in absolute terms but relative to the size of the eye itself. The iris steps back as far as its anatomy permits, leaving the maximum possible aperture for incoming light. That wide aperture is paired, in many nocturnal snakes, with a lens that sits very close to the retina, a short focal length that concentrates the gathered light onto the photoreceptor surface without allowing it to spread across a larger image plane first. Short focal length trades image magnification for brightness. The nocturnal eye is not trying to produce a large, detailed image. It is trying to produce any image at all from the faintest available signal. Most people encountering a nocturnal animal in darkness with a torch or headlamp have seen eye shine. That bright reflective glow from the eyes of cats, deer, dogs, and many other creatures. That glow comes from a tapetum lucidum, a reflective layer positioned behind the retina that bounces unabsorbed light back through the photoreceptors for a second pass, effectively doubling the chance that each incoming photon triggers a response. It is a well-established low-light adaptation found across a wide range of vertebrates. Snakes, almost universally, do not have one. The nocturnal snake eye does not produce eye shine. Point a light at a snake in darkness and its eyes return nothing. No glow, no reflection, just the dark surface of the pupil and whatever iris color is present. The snake solved the darkness problem without the tapetum, through sheer photoreceptor density and pupil geometry instead. Rod photoreceptors in the retinas of strongly nocturnal snake species reach densities that are among the highest recorded in any terrestrial vertebrate. Packing more rods into the same retinal area means more sampling points, more chances for a photon to be caught, more total signal even when each individual photon arrives rarely. This approach to low-light sensitivity through density rather than reflection is the snake's primary nocturnal strategy, and in species where it has been pushed to its extreme, it produces a retina that is almost entirely devoted to this single function. Some nocturnal snake species have retinas that are, to a close approximation, pure rod retinas. Cone photoreceptors, the cells responsible for color detection and fine detail resolution in good light, are reduced to near absence. The animal cannot distinguish colors in any meaningful sense. Its acuity in bright light is poor. It has traded away almost every visual capacity that requires cones in exchange for maximum possible sensitivity under low illumination. This is not a compromise sitting somewhere in the middle between two optima. It is a full commitment to one end of the trade-off, a retina that does one thing and does it as well as the available biology permits. This full commitment to rod-based vision also changes how the retina handles time. Rod-dominated retinas tend to favor spatial summation, pooling signals from many adjacent photoreceptors before sending the combined result to the brain. Summing signals from a wide area increases the strength of the response to dim stimuli but reduces spatial resolution, because the brain can no longer tell which specific cell within the sum pool was activated first. The nocturnal snake retina accepts this loss of spatial precision in exchange for signal amplification. It trades the ability to answer exactly where for the ability to answer something is there at light levels where a cone-based system would report nothing at all. Temporal resolution, the ability to distinguish between events that happen in rapid succession, is also reduced in strongly rod-dominated systems. Rods respond more slowly than cones, integrating light over a longer time window before generating a signal. This means a rod-saturated retina is less capable of tracking fast-moving objects in bright conditions. A nocturnal snake that hunts slowly moving mammals through nighttime vegetation is not penalized by this. A diurnal species that strikes at fast-moving prey in full sun would be. Each configuration fits its context. The relationship between eye size and reliance on vision plays out clearly across nocturnal snake species with different degrees of light dependence. Strictly nocturnal species that do most of their hunting by olfaction and chemoreception, using the tongue and vomeronasal system rather than the eyes, sometimes have eyes that are relatively smaller for their head size. The eye is present but not the primary sensory investment. Species that genuinely hunt by night vision, tracking and striking at prey using the visual system in darkness, tend to have larger eyes proportionally. Eye size, in this context, is a direct investment in visual light-gathering capacity, and the animal allocates to it roughly in proportion to how much it depends on that capacity to survive. In the species equipped with both pit organs and nocturnal eyes, some of the pit viper lineages that are primarily night active, the two systems run in parallel through the same darkness. The eye collects whatever photons the environment provides. The pit membranes collect infrared radiation from warm objects simultaneously. Both streams of information feed into the same optic tectum. The brain receives overlapping thermal and visual maps of the same space, and the combined picture is more informative than either alone. Studies examining strike accuracy under different combinations of light and pit organ availability consistently show that animals with both systems active in low light outperform those with only one system functional. Integration, in this case, is not a theoretical benefit. It is measurable in strike success rates. What is perhaps most clarifying about the nocturnal snake eye is what it reveals about the diurnal snake eye by contrast. The diurnal eye, with its flatter pupil dilation range, its higher cone-to-rod ratio, its better acuity, is not simply a better eye than the nocturnal version. It is an eye optimized for different inputs, making different trade-offs, answering different questions. Neither configuration is the correct answer to the problem of having eyes. Both are correct answers to specific versions of that problem, shaped by the particular hours and environments in which their owners have made their living across millions of years. The nocturnal snake eye, in all its rod-dense, tapetum-absent, wide-pupilled architecture, is a finely resolved response to the specific demands of darkness. It is built to work when almost everything about the environment conspires against vision, and for the animals carrying it in the habitats and hours they occupy, it works. Structure only tells half the story. What matters in the end is what an animal does with its eyes, how the optics and the neurons and the behavior all connect into something that works in the field. For most snake species, the eyes sit on the sides of the head, not facing directly forward, not angled sharply backward, but positioned along a roughly lateral axis that gives each eye its own wide monocular field covering the side and rear of the animal. The total field of view, combining both eyes, can sweep nearly the full circle around the snake's body in some species. What this configuration sacrifices is binocular overlap, the narrow zone directly ahead of the face where both eyes see the same scene simultaneously and the brain can calculate depth from the difference between the two images. In most snakes, that binocular zone is small. The visual system is organized primarily for panoramic awareness rather than precise depth measurement. Vine snakes are among the most striking exceptions. Species in the genus Ahatula and the convergently similar Oxybellus in the Americas have eyes that face considerably more forward than the typical lateral position. Paired with a horizontal keyhole pupil and a distinct rostral groove, a channel carved into the snout directly ahead of each eye, these snakes extend their forward visual field further than almost any other snake species. The groove eliminates the snout itself as a visual obstacle, giving an unobstructed forward sight line that the eye can exploit. The result is a binocular overlap zone that is wide for a snake, wide enough to support meaningful stereoscopic depth assessment when targeting a lizard perched on a distant twig. That forward binocular field matters because of where vine snakes live and what they hunt. Precise distance judgment to a small, thin target in three-dimensional canopy space, a target that may be separated from the snake by several body lengths of open air, is exactly the problem that stereoscopic vision solves well. The eyes moved forward in these lineages because the hunting task required depth information that a laterally positioned eye could not reliably deliver. Form followed function over a very long time. The science of how animals stay up with curious minds, right through the quieter hours. Many snakes that lack wide binocular overlap use a behavioral substitute for stereoscopic depth perception. Active head movement. The rhythmic side-to-side or up and down bobbing of the head seen when certain species are assessing their environment is not indecision or agitation. It is the animal generating motion parallax, the difference in apparent speed at which near and far objects move across the visual field as the observer moves. Objects close to the snake appear to sweep quickly across the visual field as the head moves. Objects farther away appear to move more slowly. The brain uses the ratio of these apparent speeds to estimate relative distance. No binocular vision required, just movement and a visual system capable of interpreting the resulting flow. It is a cheaper solution, computationally, than the neural machinery of stereopsis, and it is available to animals that cannot afford to sacrifice the wide panoramic field that lateral eyes provide. Visual prey detection in many species is initiated not in the center of the visual field but in the periphery. A snake in an ambush position, coiled motionless against bark or leaf litter, is not watching for prey directly ahead. It is monitoring the broad peripheral field for the appearance of movement anywhere in its surroundings. When motion registers in the periphery, the head orients toward it. This is a two-stage process. Peripheral detection triggers a coarse orientation response. Then the fovea-like central region of the retina, now pointed toward the target, performs the finer assessment that determines whether a strike is warranted. The strike itself, in fast-striking species, presents an interesting visual problem. The fastest snake strikes cover the distance between resting position and target in timescales short enough that real-time visual feedback cannot guide the final delivery. The neural conduction time alone, the time required for a visual signal to travel from the retina to the brain and return a motor correction to the striking musculature, is longer than the strike duration in some species. The strike, once committed, is not visually steered to its endpoint. It is aimed at a predicted location, computed from the pre-strike visual assessment, and then executed as a ballistic movement. Visual information gathered before the strike determines the trajectory. Visual information during the strike arrives too late to change it. Color discrimination, for a long time assumed to be largely irrelevant to snake predation on the grounds that most snakes are primarily motion-driven hunters, has turned out to be more present than the assumption suggested. Experimental work with corn snakes, Pantherophis guttatus, a well-studied North American colubrid, demonstrated that individuals could distinguish between objects of different colors under controlled conditions where brightness and contrast were controlled. The capacity exists. Whether it is used actively in prey detection or prey selection in wild conditions, and across how many species, remains an open and active research area. The assumption that snake color vision is vestigial or functionally irrelevant appears to have been premature. Species that specialize in taking birds show a characteristic upward orientation of peak retinal sensitivity, The region of highest rod or cone density tilts toward the upper visual field, toward the sky and the canopy above. Species that hunt along the ground or in leaf litter show the opposite, a horizontal or downward bias in retinal sensitivity distribution. The retina is not a uniform sensor. It maps the visual priorities of the animal's ecological niche directly onto its cellular geography, concentrating resources where relevant visual information is most likely to arrive. The fixed, wide-eyed expression of a watching snake, that quality of locked, unblinking attention that so many people find unsettling, is partly an anatomical consequence of the brill, which removes any blinking that might signal alertness levels. But it also reflects the limited range of eye rotation available within the snake's skull. In many species, the eye does not swivel significantly within the orbit. The snake cannot redirect its gaze by moving its eyes alone the way a human scanning a room shifts their gaze across a wide field with minimal head movement. To redirect its visual attention, the snake must redirect its head. This means that a snake that is watching something is watching it with its whole head pointed at it. A degree of directional commitment that reads, to an observer, as intense and deliberate focus even when the animal is simply maintaining a visual fix. Sit-and-wait predators, species that adopt a fixed ambush position and wait for prey to approach, tend to show higher relative investment in peripheral motion sensitivity compared with active foragers that pursue prey across terrain. The ambush hunter needs to detect the arrival of prey from any direction within its reach. The active forager needs to track a target it is already pursuing. These different hunting modes have left measurable signatures in the retinal anatomy of the species that practice them. Different density distributions, different sensitivity profiles, different allocations of the limited cellular real estate the retina offers. The eye, read carefully, reflects not just what the animal is but how it hunts. What it looks like when the eye becomes part of the snake's appearance rather than just its perception. How the eye itself is seen, camouflaged, displayed, and responded to is where the story shifts outward, from inside the skull to the surface of the animal in its world. So far, the eye has been examined from the inside, as an optical instrument, a sensory organ, a product of deep evolutionary history. Now the perspective flips. The snake's eye is also a visible object in the world. It has a color, a shape, a surface quality. It sits inside a head that other animals, predators, prey, rivals, are constantly trying to read. And the eye, like every other externally visible feature of the animal, has been shaped by the same pressures that shape the body. Not just to function well, but to appear in a way that helps the snake survive. In a substantial number of snake species, the color of the iris closely matches or complements the dominant color of the dorsal body scales. A green tree python, vivid leaf green along its back, carries a gold or pale yellow iris that sits warmly against the surrounding green, close enough in tone to avoid high contrast, different enough to refract ambient light in a way that does not make the eye the most visible point on the animal's head. A brown and tan ground boa has a similarly earthen iris. The match is not always perfect, and in many species it is clearly approximate rather than precise. But the pattern across a wide range of species is consistent enough to suggest that selection has acted on iris color for reasons that include concealment. The eye is not exempt from camouflage requirements simply because it needs to be clear. Some snake irises carry color that does not come from pigment at all. Metallic, iridescent, or mirror-like irises, the kind that produce that cool, shifting silver or gold quality under different lighting angles, arise from iridophores, specialized cells that contain stacks of thin, regularly spaced reflective platelets. Light hitting those stacked platelets undergoes thin film interference, the same physical process responsible for the colors of soap bubbles and oil films on water. The color produced depends on the thickness and spacing of the platelet layers rather than on any chemical absorbing specific wavelengths. In snakes with dense iridophore layers in the iris, the eye can shift apparent color as the viewing angle changes, appearing gold at one angle, silver white at another, faintly blue in certain light. This is the structural basis of the quality that has historically been described as the hypnotic quality of certain snake eyes. It is physics, not mysticism. The eye is often the single most visually prominent feature on a snake's head, particularly at close range. Everything else, the scales, the body pattern, the muted browns and greens of most camouflage species, is textured, broken up, designed to blend. The eye, by contrast, is smooth, rounded, reflective, and circular. Stands out. This is the problem that eye-concealing camouflage patterns solve. Running a dark stripe through the eye, from the snout, through or across the iris, continuing behind the eye toward the neck, breaks the visual outline of the eye. The circular shape that draws attention is interrupted by a linear pattern that the brain reads as background texture rather than as a feature. The eye stripe is one of the most common and widely repeated camouflage solutions in snake visual ecology. That eye stripe has evolved independently in a large number of unrelated snake families. Colubrids in North America carry it. Vipers in Africa carry it. Arboreal species in Asia and South America carry it. The convergence is not coincidental. It reflects the fact that the same visual problem keeps arising in the same form across contexts, and the same linear camouflage solution keeps being the most effective answer available from the genetic toolkit. When the same solution appears repeatedly across unrelated lineages confronting the same problem, it is a reasonable inference that the solution genuinely works. Several species, when threatened, adopt postures that specifically reduce the visibility of the eyes. Some press the head flat against the substrate, effectively hiding the eye profile against the ground. Others tuck the head beneath body coils. The behavior is not purely defensive in a physical sense. A tucked head is not better protected against a predator's strike by any obvious mechanical argument. The more plausible interpretation is visual, removing the most distinctive and recognizable feature of the head from the observer's sight line. Whether this interpretation is fully supported by controlled behavioral studies varies by species. For some it is well documented. For others it remains an inference from observed behavior. Thanatosis, death feigning, is practiced by several snake species when threatened, most famously by the hognose snakes of North America. An animal performing thanatosis goes limp, opens its mouth, and lies still. In some species, the head is oriented away from the threat during this performance. Whether this head orientation serves to hide the eye from the predator, removing the most motion-sensitive and visually distinctive cue that would signal a living animal, or whether it is simply a byproduct of the position the animal settles into, is genuinely debated. The functional explanation is plausible. The evidence for it as a deliberate visual strategy is suggestive but not firmly established. In species with warning coloration, the bright reds, yellows, and blacks of coral snakes and their mimics, the vivid patterns of certain venomous species advertising their toxicity to experienced predators, the eye is sometimes incorporated into the display rather than concealed within it. The hood markings of some cobra species include eye-like patterns, false eye spots, that may function as a threatening display when the hood is spread. These markings are on the hood, not on the actual eyes, but they interact visually with the real eyes in a way that potentially amplifies the intimidation effect of the display. The real eyes look forward. The hood eye spots face the threat. The overall visual effect of a spread cobra hood includes multiple eye-like features at once, an arrangement that behavioral studies suggest increases the freeze or retreat response in some potential predators. Albinism in snakes. The absence of melanin pigmentation due to genetic disruption of the melanin synthesis pathway produces pink or red eyes, the red color coming from the underlying blood vessels visible through the unpigmented iris. Albino snakes are well documented in captivity, where they are selectively bred for their appearance and carry no particular survival disadvantage in a managed environment. In the wild, albino individuals are rarely found and appear to have significantly reduced survival. The reasons are multiple: increased conspicuousness to predators, reduced ability to thermoregulate efficiently through behavioral color adjustment, and, particularly relevant here, heightened light sensitivity. The absence of iris pigmentation means the albino eye cannot reduce its light exposure through iris coloration, relying entirely on pupil constriction. In bright daylight, this appears to impair function meaningfully. Some species have irises with a pale silver or gold tone so reflective that, in direct sunlight at certain angles, the eye appears to produce a faint light of its own, catching ambient light and throwing it back toward the observer. This is the purely optical phenomenon that underlies the persistent cultural description of snake eyes as hypnotic or luminous. There is no bioluminescence involved, no active light production. The iridophore layers in the iris are acting as passive mirrors. What the observer sees is reflected skylight or sunlight, directed back along a narrow angle by the structured reflective surfaces of the iris. At the right angle, in the right light, it is genuinely striking. The biology that produces it is straightforward. The impression it leaves is not easily forgotten. And that impression of a gaze that appears to reach outward, to hold something in place, to exert some kind of pressure across the air between the snake and the thing looking at it, is exactly what the final chapter examines. Not from mythology, but from the neuroscience of what a fixed, unblinking stare actually does to the nervous system of the animal on the receiving end. There is one last thing the snake eye is famous for that has nothing to do with optics, and everything to do with what happens on the other side of the gaze. The idea of snake hypnosis, that a snake can hold a small animal motionless through the power of its stare alone, that the eye projects something outward that reaches into the nervous system of another creature and locks it in place, is one of the most persistent pieces of animal mythology in human culture. It appears in field observations going back centuries, in folklore from nearly every continent, and in the accounts of naturalists who watched, with apparent bewilderment, as birds and rodents sat unmoving in front of an approaching snake when escape was clearly possible. Something real is being described. The explanation for it, though, runs entirely opposite to the direction the mythology points. The freezing behavior documented in many small prey animals in the presence of a snake is not induced by the snake. It originates entirely within the prey animal's own nervous system. It is a fear response, specifically a tonic immobility or freeze response driven by the activation of threat detection circuitry centered on the amygdala and connected brainstem structures. When the amygdala of a small mammal or bird processes a sufficiently threatening stimulus, a predator close enough that the probability of detection by movement is high, one possible output of that threat assessment is complete motor suppression. Stop moving. Become still. Because for a predator that detects prey primarily through motion, stillness is a genuinely viable defense. The freeze is not paralysis forced on the animal from outside. It is the animal's own nervous system making a calculated, if not always correct, defensive choice. The snake's fixed, unblinking gaze contributes to triggering this response not through any projection of influence but through what it is: a sustained, close, non-moving attention signal from a predator. Eye contact from a predator, held without the micro-movements that social interaction typically involves, is a potent trigger for threat detection systems in prey animals. The brill, the sealed, lidless eye that cannot blink or look away, delivers exactly this stimulus continuously and involuntarily. The snake is not doing anything to the prey animal. The prey animal is doing something to itself in response to the reliable threat signal that the snake's anatomy happens to emit without effort or intent. A clarifying contrast comes from bird behavior. Many bird species that encounter snakes do not freeze. They mob, approaching in groups, calling loudly, making sustained direct eye contact with the snake while staying just outside strike range, tracking its movements closely and persistently. The mobbing bird is not paralyzed by the snake's gaze. It is doing the opposite, using visual monitoring to suppress its own flight response, keeping the threat located and assessed rather than fleeing a threat it can no longer see. Eye contact with the snake in this context is a management strategy rather than a trigger for immobility. The same gaze that freezes one species activates vigilant tracking in another. The difference is in the nervous system receiving the signal, not in the signal being sent. Work on avian anti-predator behavior, including studies on corvid and passerine mobbing responses documented in journals including Animal Behavior, has examined this contrast in detail. Controlled experimental work on cobra displays has found something worth noting carefully. When cobras spread their hood and orient toward a prey animal, the increase in the visual footprint of the snake, the hood making the animal appear larger, the eye spot markings on the hood adding additional eye-like features to the display, produces measurably longer freeze durations in some prey animals compared with a cobra without its hood raised. The display appears to amplify the freeze response beyond what the snake's face alone produces. This is a genuinely interesting finding, but it should be interpreted with care. The literature on this specific effect is limited, the experimental conditions vary between studies, and the effect size and generalizability across species are not yet firmly established. What can be said is that the display does something to the prey animal's threat assessment. What precisely it does, and through exactly what mechanism, is still being worked out. The mythology of snake hypnosis spans an unusual breadth of human cultures and historical periods. Ancient Egyptian iconography depicts serpents with a kind of ocular power. Hindu traditions include imagery of cobras whose gaze carries supernatural force. Medieval European bestiaries described snakes charming birds from trees with their eyes. Indigenous oral traditions across the Americas, Sub-Saharan Africa, and Southeast Asia include variants of the same theme. This near-universal presence in human cultural mythology is itself a biological signal. It suggests that the human response to a snake's fixed, unblinking stare, the sense that something is being projected outward from those motionless eyes, is not a learned cultural belief but a deep, conserved reaction embedded in the same threat detection architecture that produces the freeze response in prey animals. The human nervous system, encountering the snake eye, appears to be running something very close to the same ancient program. And then there is the final fact, perhaps the most clarifying one in this entire account. Snake eyes, taken across all living species, are so varied in their structure, their sensitivity, their optical design, their cellular composition, their relationship to the brain, and their ecological function that the phrase "the snake eye" as a reference to a single unified thing is, in a precise biological sense, a fiction. There are roughly 3,700 species of snake. Their eyes range from vestigial light-sensing structures barely visible beneath head scales to highly developed optical systems with fovea-like regions, forward-facing binocular fields, and retinas that have been tuned through millions of years of ecological pressure to specific hunting tasks in specific environments. Some snake eyes are rebuilt from simplified ancestors. Some retain marks of an ancient underground phase in their optics. Some see ultraviolet light. Some see heat. Some have irises that shift color in sunlight through structural interference. Some have pupils that narrow to a hairline in bright light and open to fill the eye in darkness. Not one of these is the canonical snake eye. Each is a local solution to a specific version of the problem of having eyes in a world that requires them. What the snake eye actually is, underneath all of its mythology, is a window into how biology solves problems, repeatedly, imperfectly, in parallel across independent lineages, always constrained by what came before, always shaped by what the present moment demands. Every feature of the snake eye that seems strange or alien or unsettling from the outside turns out, on examination, to be a perfectly coherent answer to a question that the animal's environment has been asking for a very long time. The eye that watches without blinking, that sees heat in darkness, that was rebuilt from simplified ruins, that carries its evolutionary history in its optics. That eye is not mysterious. It is, in the most literal sense, exactly what it needed to be. That, in the end, is what makes it worth looking at so carefully. If the science kept you company tonight, a like and subscribe means a great deal.